Welcome back to Human Biology with Dr. B. Today we get to talk about genetics and human inheritance, otherwise known as why you look like your mom and dad. So, this is a homologous pair of chromosomes. The one on the left is from your mom, the one on your right is from your dad. Now, as humans, we have 46 chromosomes. 23, a whole set, comes from your mom, a whole set comes from your dad. A homologous pair is a chromosome, one from your mom, one from your dad, that carry the same genes. So, when you get your chromosomes from your mom and from your dad, when there is a reproductive or a fertilization event, what happens is that there's chromosome 1 through 23, they're numbered. And you get chromosome 1 from your mom and 1 from your dad, chromosome 2 from your mom and chromosome 2 from your dad. And on chromosome 1, all of the genetic material on chromosome 1, whether it's from your mom or the dad, codes for the same genes. Now, a gene is a segment of DNA located at a specific site on a specific chromosome. And as we talked about, the central dogma of biology is DNA makes RNA, makes protein. And protein is the functional unit of anything in life. So the gene segment here on the chromosome is the same, codes for the same gene. For example, this is not correct information. This is just an example. So let's say chromosome 1 codes for hair color and eye color. So this gene right here would code for the protein that puts pigment in your hair or allows your hair to have a color. You do have hair and you do have hair color. It's just that from your mom, you have one gene, and from your dad, you have another. And that brings us to the discussion of alleles. An allele is an alternative form of a gene. So what does this mean? This means that my mom has blonde hair and my dad has brown hair. So I inherit from both of them the ability to have brown or blonde hair. And I have brown hair, and then my daughter has blonde hair. An allele is just an alternate form of the gene. And when we talk about alleles, we can talk about dominant and recessive codominance. We can talk about incomplete dominance and all of these things that we will discuss today. So here's your homologous pair of chromosomes. Let's talk more about alleles. It's an alternative form of a gene. Your somatic cells, which let's remember back to our last lecture, a somatic cell is every cell in your body with the exception of your gametes. Now your gametes, the whole purpose of them is to have half the amount of DNA so that when they combine, when a sperm combines with an egg, well, bam, 46 chromosomes, full person. So a somatic cell has 2 alleles for each gene. And what does this mean? That means you have one chromosome from mom and one from dad. These alleles can be the same or they can be vastly different. Now, when the copies of the allele are the same, we call this homozygous. Homo meaning the same, zygous sounds like zygote, same thing, homozygous. And when these 2 alleles on the homologous chromosomes are different, as in my case, where I get an allele for blonde hair and an allele for brown hair from my dad, they are heterozygous. And that brings us to our discussion of dominance versus recessivity. Sure, it's a word, just go with it. Now, the dominant allele, regardless of what is on the other chromosome, if there is one dominant allele, that allele will be expressed. So what does this mean? We talked about the difference between genotype and phenotype in class. Now, genotype is what is actually physically coded in your genes. So if you are a heterozygote, that means you have one dominant and one recessive allele. However, that's your genotype. However, your phenotype, you will only see the dominant effects. Now, when we are denoting this in class, the dominant allele will always be exemplified by a capital letter so that we know that dun, 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 this one is going to take over. And then a recessive allele, the only way in a recessive gene in typical Mendelian dominance will be expressed is if there's 2. And this is denoted with a lowercase whenever we're talking about the genotype and phenotype. Now, this is a great example. Dominant means that it produces a normally functioning protein. Now, does that mean that recessive, anytime that a recessive allele is produced, does that mean that it has a non-functional protein? No, it just means that it's different. For example, over here we have a very classic case of albinism. Now, albinism is recessive, and when this is expressed, when the phenotype of the recessive trait is expressed, it does not produce melanin in the melanocytes. Now, is that negative or bad? No, it just is different. And it is the expression of the recessive trait. And here's where we talk about genotype and phenotype. As I mentioned, the genotype is the set of alleles that you physically have in your genes. In your genotype, it's in your genes. Phenotype is what we see. It's the observable trait. Now, an observable trait doesn't necessarily mean how tall you are, or how strong you are, or what color your hair is, or what color your eyes are, or how long your nails are. While those are observable traits, and they are a phenotypic representation of what is in your genes, phenotype also can be as small as this protein is expressed in this way to carry out this metabolic function. So these traits look a little bit familiar because these are the ones that we talked about in class in lab last week. So freckles is skin pigmentation. No freckles is recessive. Freckles is dominant. Unattached earlobes is dominant. Attached earlobes is recessive. Down here, widow's peak is dominant, and a straight hairline is recessive. And then tongue rolling is dominant, whereas not being able to do that is a recessive trait. So how do we express dominance and recessivity, and how do we have this much diversity in our population? Well, according to Mendelian genetics, Mendel is the monk in the 1800s who actually discovered dominance and recessivity. We have a number of, quote, laws that help us understand where the diversity comes from. So the first one is the law of segregation, and this basically says alleles separate during meiosis, and this occurs in meiosis 2. The law of independent assortment, as we talked about, is meiosis 1, more specifically anaphase 1, where homologous chromosomes line up randomly and then are pulled to the opposite sides of the poles before we move through our second division in meiosis. And then we have the law of dominance. And this says when there is one gene that is dominant, it will be expressed, whereas the recessive gene will only be expressed if there are 2 copies of it. And the way that we show this or draw this is through a Punnett square. Now, a Punnett square is nothing more than a diagram that we use to predict what the outcome of a certain type of cross will be. So up here on the top, we see one individual and we see the second individual over here. And we are going to predict what color eyes these 2 people, should they have a reproductive event, we'll call it that, what the chances are that they will have a blue-eyed child. Now up here, so each person has a mom and a dad. So let's say that this one came from mom, this gene came from mom, and this gene came from dad. Remember, it's the same gene, it's on the same chromosome, different alleles. So brown basically means pigment, there's pigment in your eyes, and blue codes for no pigment in your eye color. So what happens is here we have 2 heterozygous parents. So we bring the large B down and put it in each box here. We bring the large B across here and here, the little b across here and here, and the little b across here and here. And when we look at this, we can predict that it's most likely that this couple has a 75% chance of producing a child with brown eyes. However, there is a 25% chance of producing a child with blue eyes. Because look here, this is a homozygous dominant. This will have the dominant phenotype. This is heterozygous, but because we have one dominant allele, the dominant phenotype will be expressed, same here. And then this is the only recessive, homozygous recessive, and this is the only opportunity for that recessive trait to be expressed. So Our phenotypic ratio is 3, 1, 2, 3 to 1. Our genotypic ratio is 1 to 2 to 1. And here we can see this again. Oh, I love it. Here we have eggs and sperm. This is so funny. So this guy in his sperm is only going to contribute the recessive allele. This beautiful young lady will contribute the dominant allele. And then if she is homozygous dominant for skin pigmentation, then the only thing she can contribute is the dominant allele, which means that all of their children will have the dominant phenotype, despite the fact that they are heterozygous. Because remember, with dominance, anytime there is the presence of the dominant allele, that will be expressed. So this is what we call a monohybrid cross. So this is a cross where both parents are heterozygous. A monohybrid cross is where we look at the heterozygotes in both cases for each parent and what the chances of their children will be to have various phenotypes. And anytime we do a monohybrid cross, it is a heterozygote plus a heterozygote, and every monohybrid cross, when we are dealing with straight dominance, is a 3-to-1 phenotypic ratio, because heterozygote, heterozygote, 3-to-1. The genotypic ratio every single time will be 1-to-2-to-1. Now, a dihybrid cross, this is what this is called, a dihybrid cross is where we look at two traits. They're on different chromosomes, two traits on different chromosomes to see how they are inherited. Now, in this case, we're looking at hairline and skin pigmentation. Now, for a dihybrid cross, that means each individual is heterozygous for both traits. So here we can see heterozygous for skin pigmentation or freckles, heterozygous for hairline. Same over here. And then when we go through the Punnett square, now keep in mind the way that this dihybrid crosses are very confusing for students, but keep in mind that every single egg and sperm has to have a gene from each chromosome. So each egg and sperm has 23 chromosomes. Each egg and sperm, every single chromosome, which means it has every single copy of the genes. So in a dihybrid cross, that means these genes that we are looking at are in different chromosomes. So when we draw this out, we need to make sure that every single sperm has each chromosome. And so in this instance of a heterozygote, we separate them accordingly. So dominant, big F, big W, big F, little w, little f, big W, little f, little w. So we have all the possible combinations for these genes in these sperm and eggs. So we put them all right here. And we do the same thing with the monohybrid cross. We put the big F and big W here. Big F, big W, big F, big W, big F, big W. And we pull them all the way down. And then we take these genes and we pull them all the way across. And we always put the genes that are on the same chromosome, for example, F and W, we put them next to each other so we know what the outcome could possibly be. Now, when we do a dihybrid cross, the phenotypic ratio will always be 9-to-3-to-3-to-1. Memorize that. A dihybrid cross, the phenotypic ratio is always 9-to-3-to-3-to-1, every single time. And here are the options. Now, a pedigree. Every time I hear the word pedigree, I think of dog food. However, in genetics, a pedigree is basically a roadmap showing you generations of individuals, generations within a family, and how the genes have been inherited. So based on the phenotype, and we actually did this without drawing a pedigree in lab the other day, because when we were trying to figure out your genotype, we thought back to your parents and what their phenotype would be. And then we can know, based on what parents and grandparents express, what could potentially be the offspring's phenotype and genotype. So, with this, we look at it's very important for determining autosomal dominant and autosomal recessive genetic disorders. And this is what it looks like. Basically looks like just a lot of circles and squares. Now, a clear or a white circle is an unaffected female. A white square is an unaffected male. And then blue circles and squares are affected, meaning they are expressing the gene of interest. And in this case, you can see that the dominant trait will express it. And so, yes, pedigree of a dominant trait, pedigree of a recessive trait. So here you can see that each parent is a carrier. We produce heterozygotes, well, just here. Mary's a heterozygote, and then we have the chance of 50% of the children being affected. And so every time there is a crossbar right here, that means it's a reproductive event. So here, and then these are the offspring, as well as these 6 offspring from this pairing. So now that we've just covered dominance, let's kind of throw a wrench in the situation and talk about codominance. This is where both alleles produce functional proteins. Okay, so think back to dominance. In the recessive homozygote, it produces a completely different protein that does not allow for the expression of that particular trait. Hair color or skin color, for example, doesn't allow for the production of melanin if it's recessive for that trait. So in codominant alleles, that means everybody is expressed. And the best example of this is AB blood. Now, I will draw this Punnett square in class so I can show you better, seeing as though my drawing isn't working currently in technology. But when blood types are determined, it produces a polysaccharide on the red blood cell. And that determines whether you're A or B or AB or O. O meaning you produce no polysaccharide. A meaning you produce the polysaccharide. B, you have the B polysaccharide. And AB, you produce both. But when you have codominance, everything is expressed. And incomplete dominance is where the gene is somewhat in between the homozygous dominant and homozygous recessive. So the best example that they have here is sickle cell anemia. So when there is, so this is the heterozygote, and it's somewhere in between homozygous recessive and homozygous dominant. And so the cells, the red blood cells, have an abnormal shape, but not enough to cause problems. Whereas in sickle cell anemia, number one, this does not allow for fully oxygenated tissues, as well as can cause clots very rapidly. Another example of this is white, red, and then pink flowers. The pink is halfway in the middle. So it expresses the red, but it also expresses the white. And so that allows for the whole range of red, pink, and white flowers. And then sex-linked traits, we covered this a lot in class already during lab. Now, when they can be sex-linked Y or they could be sex-linked X traits. And sex-linked X traits are passed down through the mother. And in males, all sex-linked X traits will be, if the female is a heterozygote, a carrier, or a homozygote, whatever is on the X chromosome in a male will be expressed. Now, females, as we talked about, are determined in the absence of a Y chromosome. And then males are determined by the presence of a Y chromosome, which can only be given by males. A male will express the X-linked gene even if they are recessive simply because they only have one X chromosome. A female will only express it if she receives 2 of the recessive trait, one from mom and one from dad. And then X-linked expression is much more common in males than it is in females. And some great examples, color blindness, muscular dystrophy, and hemophilia. We talked about color blindness in class. It was very fascinating. Here you can see this. So the carrier over here, here's a carrier mom, and she can contribute an X and an X to her eggs, whereas a father contributes an X and a Y. So in this, the mom has a normal phenotype but is a carrier. Therefore, half of her boys could potentially have or express the recessive phenotype. And in this case, we could use color blindness or anything that we talk, hemophilia, muscular dystrophy, etc. So if the mom is a carrier, that means that half of all males will express it and none of the females will express it unless the dad also has it. If the dad has it, that means that one female will be a carrier and the other will absolutely express it. So now you should be able to define and differentiate between gene, allele, dominant, recessive, homozygous, and heterozygous. You should be able to draw and apply the Punnett square, use a pedigree, and apply the principles of codominance, incomplete dominance, and sex-linked traits. Thanks for joining me again for Human Biology with Dr. B. See you next time.